Thank you, the chair, for the introduction. So first of all, I would like to thank the Institute Pasteur Korea and also Sujin for having me here to share our research. Of course, as the chair introduced, I wear a lot of hats. So probably I'm now currently representing the Hong Kong Pasteur Research Pool formally because this is a Pasteur Network meeting. So before I jump into my discussions, I always ask these questions, in my AMR talk. So last year, I also asked this question: how many of you have never taken antibiotics in your lifetime? I'm sure, none of us. If you are the one who never take the antibiotics in your lifetime, I think you probably come from Amazon jungle or some, untouched tribe. So definitely, the answer could be a 0. So this is also indicating that our modern lifestyle live with the antibiotics. We cannot live without antibiotics because antibiotics is also a powerful modern medicine for us. But we are using antibiotics in a little bit of wrong way. So of course, sometimes we use the antibiotics as a prophylaxis, sometimes very amputative, preemptive kind of prescription of the antibiotics, even we don't know what is the causal organism. But even we know the causal organism, we try to use the more directive or definitive use of the antibiotics. And also, sometimes we try to suppress the antibiotics to suppress the bacteria for long term. So we use those kind of antibiotics in a longer time, such as in mycobacterial tuberculosis infection. So anyway, we are using antibiotics in many ways. Antibiotics become a candy for us. So that is the problem. Because Sir Alexander Fleming already indicated that, who is the first discovery of the antibiotics. He mentioned that antibiotics resistance will come if we are using it improperly. So antibiotics resistance is not a very new topic. It's already indicated, in the Nobel lecture of the Sir Alexander Fleming. At the time, the antibiotics has just discovered. So why we are now having, why we are talking about AMR nowadays? Because we are using this improperly, and that's why the collateral damage, which is AMR, is happening. So this slide has been used by many speakers, and I think everybody familiar with that. So Sir Jim Monod, who is the economist, made this prediction report, and indicated that there will be around 10 million, deaths attributed to AMR in 2050. So at the time when I read this, report, I'm always have a question. It is true. Can it be possible? However, just recently, the Lancet report of the global burden of AMR indicated that it is already been 1.2 million, 1.27 million deaths already. So we are not far from that 10 million. This is also, alarming to us. AMR is a silent pandemic, what Sujin mentioned in her presentation. So we need to tackle now. However, AMR is so complicated. It's a very complex issue because there are many driving factors, make AMR. It can be because of misuse of antibiotics. It can be because of the, agricultural use of antibiotics. And it can be also contributed by the environment and also wild animal. So this, of course, that's why nowadays we are trying to use the One Health approach to tackle this AMR issue. So, in addition to this, many driving factors, AMR is even further complicated because it can be the issue of the transmissions in the health institutions or in the hospital. It can be transmitted from one person to one person within the population. But it can also be the transmission through the gene level, plasmid level, what one of the speakers this morning indicated that. So multiple layers of the transmission can be possible, and that is making AMR very difficult to tackle. So, but WHO is very smart when we have a lot of target, a lot of complication. They try to make it simple. So in 2016, they released this 12 drugs-resistant bacteria as a priority list in order to tackle AMR. So of course, you can see in the critical priority list, most of them are carbapenem-resistant bacteria. So we have to also admit that this carbapenem-resistant bacteria are giving a huge threat to our healthcare institution. So this is also Lancet, global burden of report. And in this report, we also found that there are many bacteria that are giving a burden for AMR. However, the first 3 bacteria that you can see that is E. coli, S. aureus, and Klebsiella. So Klebsiella pneumoniae is one of the AMR bacteria that can be resistant to the carbapenem and also the next generation cephalosporin and also extended spectrum beta-lactamases. So this is one of the targets that I would like to emphasize today. So when we look at the global distributions of the carbapenem-resistant Klebsiella pneumoniae, you can see that different parts of the world have facing a problem with this bacteria. But it's very difficult to tackle this, carbapenem-resistant Klebsiella pneumoniae. Because in traditional microbiology, when we are, study, we only focus on Klebsiella pneumoniae. But recently, this bacteria also expanded the family member into the Klebsiella quasi-pneumoniae complex, and you can see that this Klebsiella quasi-pneumoniae has been reported in many countries, in the clinical patients. So it's indicated that we should not only focus on Klebsiella pneumoniae, but we also have to focus on the other Klebsiella species, such as Klebsiella quasi-pneumoniae. However, trying to do the surveillance on the AMR bacteria are very challenging because there are too many back-drugs combination. Okay, so we have so many bacteria, so many antibiotics. If you're combining those things, there's tens. Where to focus? The focus is a big issue in the AMR field. And there are too many sample types. It can be, AMR bacteria can be found in the respiratory tract, also in the gastrointestinal tract, and also other body conditions. And also, different layer of sampling. Whether we want to do the sampling in the community level, or household level, or even individual level, how those data could be representative enough in order to make the policy. This is always a question in the AMR surveillance. There are also too many hotspots because of the One Health approach. So when we are talking about even human sector, there are many hotspots for AMR. But when we include the environment and also agricultural sector, there are many other hotspots as well. Of course, that's why when we are doing this surveillance for AMR, it is very costly, and technically and logistically, it is a huge burden. So this paper indicated that environmental hotspot is really a big target for the ARG. And this is also true, every day we are going to toilets and we are just pushing out all our waste into the, environment. So that's why probably environmental, surveillance could be one of the approach to tackle or to monitor the AMR. So that's why we want to use this approach. And this, systematic review also indicated that the anti-AMR surveillance using the wastewater is really meaningful. You can see the correlation between the phenotypic and also genotypic between the environmental sample and also the clinical sample. They are highly correlated. So definitely we can use the environmental surveillance to monitor what is happening in our healthcare institution in regard to the AMR. This is our study we have done during the COVID-19 in Hong Kong. Hong Kong is very famous in fighting against COVID-19 through using the wastewater-based epidemiology. So I was one of the members in this team to use the wastewater epidemiology to monitor the epidemic wave, in Hong Kong through the COVID-19 pandemic. We successfully predict the kind of more recent pandemic wave, and then we can take care and also do the public health action. So this is one of our successful story. That's why we think probably we can also use the wastewater-based epidemiology in AMR field. And I am one of the expert panel member in order to produce this very, historical and remarkable, document called a One Health Priority Research Agenda for the AMR by the quadripartite, including WHO, OIE, FAO, and also UNEP. So if you want to know more detail about what are the research agenda for One Health AMR, please go and read this document. But I would like to emphasize two things. So one thing is the about the wastewater could be a solutions in order to, monitor the development and circulations of AMR in One Health sector. Another kind of quote I would like to bring from this document is whether we can use the AMR hotspot proactively identify and mitigate using a One Health approach. So this is the two major, point that is related to the wastewater epidemiology from this document. So with the aim of this, we try to conduct a longitudinal monitoring of the AMR in a hospital wastewater discharge line as a pilot project. So we conducted this project in one of the hospital called Queen Mary Hospital in Hong Kong. It's a teaching hospital. It's a tertiary level hospital. And we collect the sample weekly, from the hospital wastewater and also monthly from the downstream municipal wastewater treatment plant in order to monitor the AMR and also ARG in the sample. So this is how we collect a sample. You can see that, we wear all this PPE in order to collect the sample. It's very tedious work, even we collect weekly sampling. But in the future, we are now developing a technology using the automated, sampling approach so that we can be easier to have a kind of more real-time monitoring system in the future. But I would like to emphasize the sampling sites. So we identified 3 sampling sites in the hospital. The first size is the office size. So the office size is not related to any clinical work, but another 2 clinical sites. The clinical site is related to a lot of ICU. But clinical size B is just related to some pathology or gynecology department. So we try to compare, the sample from these 3 different sites. So this is also another wastewater sampling at the downstream wastewater treatment plant, how we did it. So after we collect the sample, we try to isolate the bacteria, and also we try to do the phenotyping and genotyping, and also the resistant profiling on that. It's very simple kind of workflow. And for our kind of monitoring, we found that we did both AMR, ARG, as well as also the residue analysis. Based on our residue analysis, you can see that it's office quarter. We didn't see antibiotics a lot. But in the clinical works, the wastewater came out from clinical work, there are a lot of antibiotic residue. And that is also correlated with a lot of bacteria, and that is associated to multi-drug resistant. Sorry, because of maybe some issue with the figure, it didn't show properly, but the red color show the multi-drug resistant bacteria. And we also try to look at more detail on the different sites, and then you can see that the clinical site A have a bloom of the Klebsiella species, and that is related to the ICU. So that is also indicated that probably there is some related to the Klebsiella related outbreak or even incidences in this hospital. So we also try to look at the different antibiotic resistant data. We found that this clinical site A is only uniquely resistant to meropenem, not in other sites. And we found that this is very significantly, distance from other sites, and it is very important. So we try to deep down on this Klebsiella isolate collected from the clinical site A. And these are the isolate that we analyzed based on their whole genome data. And we found that they are not Klebsiella pneumoniae, but our multi-TOF analysis indicated that they are Klebsiella pneumoniae. But based on our whole genome sequencing analysis, most of them are Klebsiella quasi-pneumoniae. And we also found a few MLSD, like novel MLSD type in this, isolate. Predominantly, these isolate carry the BLA-GES5 gene, in order to be resistant to the carbapenem. And we are very interested on this phenomenon, and we try to look at other global isolate about the Klebsiella quasi-pneumoniae. And we found that isolate from our study only carry the BLA-GES-like gene, no other isolate collected in the different country. So this is also indicating that there is an emergence of this new genotype of the AMR bacteria. And you can see that they also carry quite a lot of plasmid replicon. So I'm sure that one of the speakers this morning will be very interested in this isolate. So they carry a lot of plasmid. And we could also see that the number of plasmid and the number of ARG are highly correlated. If they carry more plasmid, they carry more AMR genes in their genome. And then we try to analyze whether these isolates can share their resistant gene, which is the BLA-GES5, into other isolates or not through horizontal gene transfer. So we constructed the phylogenetic analysis based on their BLA-GES5 gene and the flanking region. And we found that most of the isolates, they carry this kind of gene cassette, either upstream or even downstream of the BLA-GES5, which is the integrase gene. This is a mobile genetic element. So it's indicated that these genes can be genetically horizontally transferred to other bacteria, but the mechanism of the conjugation could not be proven through the experimental research. We found that they are not conjugation, but they can be through this integrase mobile genetic element to move to the other bacteria. So this study has been published already. So if you have interest more deeper on this study, please go to read this paper. Of course, sometimes people say, oh, this is whole genome sequencing analysis. How about metagenomic-based, monitoring approach? So we also conducted in this study using the metagenomic base. So this is our workflow. It's very easy. And based on our data, we also found that the BLA-GES5 gene is significantly higher in the clinical site A, which is here. You can see. So it's indicated that this is one of the emerging problems, And this gene is also transmitted down to the treatment plants. And if we cannot, monitor or, screen out these genes in the treatment plant, definitely it will go to the wider environment for sure. And it can be also transmissible to other bacteria. So that's why we think this is a very important message. But in this metagenomic study, we also try to measure the ARG number. So this is the log-adjusted ARG count per genome. And then we also try to correlate with the temperature of the sample. And we found that they are significantly correlated. So a lot of people say the AMR could not be related to the climate change. But actually, based on this data, perhaps global warming can also enhance the number of the ARG in our environment. That is also a very good message, and we have to tackle this. So I just want to summarize my presentations by this 4 points. So we all identified that the carbapenem-resistant Klebsiella quasi-pneumoniae from the Hong Kong wastewater, especially from the hospital wastewater. And this is very important. And they are carrying this new genetic feature called BLA-GES5, and that is also making the resistant to carbapenem. And there is a potential horizontal gene transfer even through the mobile genetic element that we need to monitor in the future. So of course, I would like to kind of promote and also indicate that wastewater-based epidemiology is very useful to monitor the emergence and spread of the AMR bacteria as well as the ARG. We are now trying to expand this project or similar type of project to other countries that we are collaborating, which is in Thailand, mainland China, and also Cambodia as well. So hopefully in the future, we can use the wastewater-based epidemiology as a tool, as a cost-effective tool in order to monitor the emergence and also spread of the AMR in the community. With that, I just want to thank all the team members from my lab. My lab name is System Microbiology and Antimicrobial Resistance Lab called Smart Lab. So, and all the funding agencies that support my research and team. Thank you so much.